Hello everyone, welcome back to my channel. In today's session, I will be discussing on unit genetics, that is chapter 5 and chapter 6. Watch till the end because many questions have been discussed in this session. So let us start. Question: which of the following was not proposed by Mendel? And it is option number 3, incomplete dominance. Next question: the two allelic genes of a pair are located on, and it is option number 2, the homologous chromosomes. Next question: a gamete normally contains, and it is option number 2, one allele of a gene. Next question: checkerboard, which is the graphical representation to calculate the probability of all probable genotypes of offspring in a genetic cross, was developed by, it is option number 2, Reginald Punnett. Next question: two pea plants were subjected to cross pollination. Of the 183 plants produced in the next generation, 94 plants were found to be tall and 89 plants were found to be dwarf. The genotypes of the two parental plants are likely to be, and it is option number 1, capital T and small t. That is tall and dwarf. Next question: when a hybrid pea plant for yellow round seeds, that is capital Y, capital R, small r, is self-pollinated, the phenotypic ratio in the next generation would be, that is option number 2, 1 is to 2 is to 2 is to 1 is to 4 is to 1 is to 2 is to 2 is to 1, which is the dihybrid genotypic ratio. Next question: a cross between F1 hybrid and the recessive parent is called, it is option number 4, test cross. Next question: test cross is a cross between, it is option number 2, F1 hybrid and the recessive parent. Next question: the genotype of a plant showing the dominant phenotype can be determined by, it is option number 1, test cross. Next question: to determine heterozygosity and homozygosity, a plant must be crossed with, it is option number 1, recessive plant. Next question: a cross between two plants, one having 24 centimeter long internodes and the other having 12 centimeter long internodes, produced F1 hybrids, all having 18 centimeter long internodes. This is a case of, and it is option number 2, incomplete dominance. Next question: in Anthedium or Snapdragon, true bred red and white flowered plants are crossed. In F2 generation, they form, and it is option number 4, 25% red, 50% pink, and 25% white flowered plants. Next question: in monohybrid crosses, absence of complete dominance is indicated by the F1 plants that exhibit intermediate characters. This can be further confirmed if the phenotypic ratio in F2 generation is, and the option is 3, that is 1 is to 2 is to 1. Next question: the F2 generation offspring in a plant showing incomplete dominance exhibit, the option is 4, that is similar genotypic and phenotypic ratios of 1 is to 2 is to 1. Next question: in incomplete dominance, genotypic and phenotypic ratio are the same. It is option number 3, 1 is to 2 is to 1. Next question: haploid diploid sex determination system is seen in, and it is option number 4, honeybees. Next question: among honeybees, the option is 1, males are haploid and females are diploid. Next question: females are heterogametic in, and it is option number 3, birds. Next question: males develop parthenogenetically in, and the option is 4, honeybees. Next question: two lines between the male and female in a pedigree chart indicates, and the option is 2, male and female are related to one another. Next question: sickle-celled anemia is caused due to the substitution of, and it is option number 2, glutamic acid at 6th position of beta chain of hemoglobin by valine. Next question: an example for an autosomal blood disease is, and it is option number 4, thalassemia. Next question: in thalassemia, it is option number 1, there is reduced synthesis of alpha and or beta chain of hemoglobin. Next question: alpha thalassemia is caused due to, it is option number 1, mutation or deletion of HbA1 and HbA2 genes on chromosome 16. Next question. The number of chromosomes in an individual suffering from Klinefelter syndrome is, it is option number 4, 47. Next question. An extra X chromosome in human beings causes, it is option number 2, Klinefelter syndrome. Next question. An extra autosome in human beings causes, and it is option number 3, Down syndrome. Next question. An additional copy of chromosome number 21 causes, and it is option number 3, Down syndrome. Next question. Gynecomastia is a condition seen in, and it is option number 2, Klinefelter syndrome. Next question. Down syndrome or Turner syndrome or Klinefelter syndrome is due to option number 4, non-disjunction of chromosomes. Next question. Nucleic acid was first isolated from pus cells by, is option number 1, Friedrich Mischer. Next question. The distance between the two strands of DNA is, it is option number 1, 20 angstrom. Next question. Each nucleoside consists of, and the option is number 1, a base and a sugar. Next question. The two strands of DNA are linked by hydrogen bonds established by, it is option number 1, adenine bound with thymine, guanine bound with cytosine. Next question. In a DNA molecule, it is option number 1, A plus G is equal to T plus C. Next question. In the diploid content of human DNA, there are, it is option number 4, 6.6 into 10 to the power 9 base pairs. Next question. A nitrogenous base in DNA is linked to the pentose sugar through a, it is option number 3, N-glycosidic bond. Next question. The nucleotides in DNA are linked through 3' to 5', and the option is, it is 4, phosphodiester bond. Next question. The backbone in a double-stranded DNA molecule is formed due to, it's option number 1, deoxyribose sugar and phosphate. Next question. The pitch of the helix of a DNA molecule is, it is option number 2, 3.4 nanometer. Next question. Information transfer from RNA to DNA is called, it's option number 4, reverse transcription. Next question. Information from the nucleus to the cytoplasm is conveyed by, it's option number 2, mRNA. Next question. The adapter RNA or soluble RNA molecule is, it's option number 2, tRNA, that is transfer RNA. Next question. The experimental proof of semi-conservative replication of DNA was provided by, it's option number 3, Messelson and Stahl. Next question. Messelson and Stahl used dash for their experiment to show semi-conservative replication, and the option is 2, E. coli. Next question. During DNA replication, Okazaki fragments are found in the lagging strand so as to, the option is 2, facilitate the functioning of DNA polymerase from 5' to 3' end. Next question. DNA replication is semi-conservative because, it's option number 3, one of the strands in each molecule synthesized is parental and the other strand is new. Next question. Hershey was awarded Nobel Prize in 1969 for providing, it is option number 4, semi-conservative DNA replication. Next question. Central dogma of protein synthesis is a flow of information from, it is option number 3, from DNA to RNA, RNA to proteins. Next question. A transcription unit in DNA is defined primarily by the following 3 regions in the DNA, and the option is 1, a promoter, a terminator, and the structural gene. Next question. rRNA, that is 28S, 18S, 5' is transcribed by the enzyme, and the option is 1, RNA polymerase 1. Next question. hnRNA, that is heterogeneous RNA or precursor mRNA, is transcribed by the enzyme, and the option is 2, RNA polymerase 2. Next question. tRNA or 5S RNA or snRNA means small nuclear RNA is transcribed by the enzyme, and the option is 3, RNA polymerase 3. Next question. The region or sequence of transcription unit that undergoes transcription is, the option is 2, structural gene. Next question. During transcription, Capping refers to the addition of, and the option is 4, methyl guanosine triphosphate at 5' end. Next question. During protein synthesis, AUG functions as the initiator codon in mRNA. What should be the anticodon on the tRNA molecule that picks up methionine specified by this codon? And the option is 1, UAC. Next question. The sequence of events mentioned below are symbolized by alphabets. Choose the correct answer where the alphabets are matched with the processes. So the option is 1. A. Reverse transcription. B. Replication. C. Transcription. D. Translation. Next question. All the terminator codons begin with the nucleotide and the option is 2. Uracil. Next question. The function of UAA, UGA or UAG nonsense codon is to. Option is 3. Stop protein synthesis. Next question. The terminator codons or stop codons are, it is option number 3, UAA, UAG and UGA. Next question. The largest known human gene dystrophin contains, it's option number 2, 2.4 million nucleotide bases. Next question. The technique of DNA fingerprinting was initially developed by, its option number 2, Alec Jeffreys. Next question. The technique of DNA analysis which is used in forensic science is, its option number 3, DNA fingerprinting. Next question. Tandem repeats, that is VNTRs, in the DNA molecule are highly useful in, it is option number 2, DNA profiling. And the last question of the session, a technology which has found immense use in solving cases of disputed parentage is, and it's option number 2, DNA fingerprinting. Thank you for watching my videos. Do not forget to like, share, comment and subscribe. Stay tuned for more such videos.